In this video, we're going to be discussing our next sense, which is eyesight and vision. We start off by drawing an eyeball, and the focus is going to be on discussing the structures of the eye through which photons of light must pass. And so the focus that we're going to be discussing here is going to be on the clear structures of the eyeball. So we're not going to be labeling very much here, but I will kind of discuss things as they are relevant. At the very anterior of the eye, which is to the left in this picture, we have the clear cornea of the fibrous tunic. The cornea, sort of the most anterior structure of the eye. And then behind that we have the anterior segment, which is filled with aqueous humor. And I've invaginated it here to show you where the iris would be. The iris, of course, is pigmented and solid, but the pupil in the center there is going to be clear. So the aqueous humor is clear, and the pupil is a hole through which a light can pass. And behind that we have the ciliary body is our next invagination, and the little strings shown there are suspensory ligaments, which attach between the ciliary muscles, the ciliary body, and the lens. Now what I want to do is I want to show that photon of light starting to come through all of these structures that we have drawn. We start off going through the cornea, aqueous humor, pupil, and lens. Then the photon of light progresses through the posterior segment, which contains vitreous humor, which is also clear. And we're going to draw the photons of light going all the way to the back of the eye. And you notice that there is refraction of the light, or a bending of light, when it comes to certain structures. So the refraction of the light is going to be most apparent as it passes through the cornea, through the aqueous humor, and then through the lens. And the hope is that all of these photons of light, these lines, are all going to be kind of uniting together at one point at the back of the eyeball. So you'll see that even though they are scattered to begin with, and then they continue to scatter a bit, they eventually refract to exactly the point where we want them to refract to, which is our nice central focus point, all the way to the back of the retina. So light passes through all of these clear structures all the way to the back of the eye, where it is finally sensed. I'm also going to be drawing the optic disc, which is a structure that does not have any kind of cells, which leads to cranial nerve 2, or the optic nerve. Now we are going to draw the cells that are all the way back here. This is the structure of the eye called the retina. The retina is actually filled with 3 layers of cells. There's actually 5 cell types here, but there's 6 cell types, but there's 3 layers, so we're going to draw all 3 layers. In blue, we're going to go ahead and draw our ganglion cell layer, and you'll notice that the axons of the ganglion cells actually become the cranial nerve 2, or the optic nerve. Behind that, we're going to draw a layer of black cells. These are going to be bipolar cells. And all the way to the back, all the way to the posterior of all this stuff, are going to be our photoreceptors. Our rods are going to be drawn in brown, and our cones in green. Now what I want to do is I want to take that retina and turn it out so that we're actually seeing it face on, so that we can see some of the structures here. First of all, we're going to go ahead and label our macula lutea, and the hope is that we have the photons of light, which we're showing with these kind of red squiggly lines here, are going to hit right on that macula lutea. It's also important to draw, and I'm just going to draw an outline of it here, the optic disc, which is that structure through which the cranial nerve 2, the optic nerve, actually leaves out of the back of the eye. Remember that there's no cells where the optic disc is, where that cranial nerve 2 leaves the eye. Now I'm going to go ahead and label the macula lutea, that central point, and right in the center of the macula lutea is actually the fovea centralis, which literally translates as the central focal point. And what we're going to do is we're going to go ahead and draw in green dots. We're going to draw all of our cones. Most of the cones of our eyes are going to be located right here in the macula lutea, and especially in that fovea centralis. There will also be some cones outside of there in what we call the periphery, or the peripheral vision area of the eye, of the retina. If you're curious about the numbers, there's about 6 million cones found, mostly inside of the macula lutea, and about 200,000 of them are going to be found in that fovea centralis, which is a very, very small area, about less than a tenth the size of the macula lutea, but it has about 200,000 cones. Now what I want to do is I want to actually draw a cone. The cone has kind of an unusual structure. The way that we're drawing it is we have 3 different short little axons there that are going to be sending the signal along. It kind of looks like a carrot, like a short carrot. And I chose to draw this in green, but it could be drawn in pretty much any color. It typically is drawn as a color because it does see color. Now we're going to add brown. What we're going to see is that there's a few brown spots right in the center in that macula lutea, but most of the brown, which we have said are rods, are going to be outside of here, outside of the central vision. Whereas there are 6 million cones, mostly in the macula lutea, there are 120 million rods, mostly outside of the macula lutea. Of course, they are not going to be in the optic disc, though. There are no cells in the optic disc. There are no cells in the area where the optic nerve leaves the eye. And let's go ahead and draw our rod. So you'll notice that the rod isn't quite a carrot anymore. It's more of a, well, it looks kind of like a rod. It's a longer structure here, but there's some similarity there. So the cones and the rods. The cones are responsible for central vision. They also are able to see colors. There's 3 different types of cones. And interestingly enough, and I'll kind of show this later, the cones are responsible for high acuity vision, the ability to see things very clearly and acute. Now rods are different. They are responsible for peripheral vision rather than central vision. Remember, the cones are found in the macula lutea. The rods tend to be found in the periphery there. The rods tend to be very sensitive. They don't tend to be high acuity, but they do tend to be high sensitivity. I'll show that as well. Another thing to say about rods is that they do not see color. It's not so much to say that they see black and white. It's more to say that they're either on or off. They either see light or they don't see light. So as you see, we drew 3 rods in a row, and then we're going to draw 1 more cone off to the side here. And then we're going to begin to build our next layer of cells, which are going to be the bipolar cells, which, as we labeled earlier, are going to be in black. One of the things to note is that each cone has its own bipolar cell. We do have 3 synaptic terminals here, or 3 synapses, but the bipolar cell has simply 3 receptors, or 3 receptive regions, and 3 dendritic regions, if you will, as far as when we're talking about cones. So for the cones, each of them has its own bipolar cell. This is important for acuity. Now you'll notice that the story is a little bit different for the rods. For the rods, the bipolar cells tend to not be as 1-for-1. Let's show you what I'm talking about here. So for this first one, the first rod that we're drawing here does have its own bipolar cell. However, the second and third rod here actually share a bipolar cell. You'll notice the synaptic connection is receiving information from both. We're going to go ahead and label our photoreceptors here, which are our rods and our cones. And we're going to go ahead and label our bipolar cells as well in black. And then we're going to go ahead and use the color blue to label our ganglion cells. The ganglion cells, you'll notice, there's going to only be 3 that we're drawing here. One of the ganglion cells is going to be receiving information from one cone, 1 ganglion cell from the other cone, and then only 1 ganglion cell is actually going to be receiving information from the 2 bipolar cells that were associated with those rods. All of the ganglion cells are going to send their axons in the direction of the optic disc, in the direction of cranial nerve 2, the optic nerve. So again, notice that each of the cones has its own bipolar cell and its own ganglion cell, and therefore each time that a cone receives information, it receives a photon of light, it's going to have a very specific focus. Yes, it's going to have a very specific way of sending exactly the information about that photon of light. in that particular locus directly to the brain. So let's go ahead and draw a photon of light here. You'll notice this photon of light is hitting the cone on the left and a little bit of that rod on the left. And then we'll do a second photon of light, part of which is hitting each of the rods a little bit. So let us say that there is enough photons to cause changes within the pigments inside of the rods and the cones in these cases. Well, that cone on the left-hand side, that first cone, is going to be sending extremely specific information about just the photon of light that hit it. Whereas the rods in the center, it actually doesn't matter if the photon of light hit the first, second, or third, or if it hit 2 over the 3 or all 3 of the 3, the signal is going to still appear the same. So that one ganglion cell that is drawn in the center here receives information from all 3 rods, and it's really just going to be the same whether it's, a strong photon series of photons of light, and no matter what location it is amongst those 3, it's still going to appear to be the same signal. This is why rods tend to be more sensitive. A photon of light that's hitting way off to the periphery will still have just enough oomph to allow for a bipolar cell to receive the signal and a ganglion cell to receive the signal as well. Whereas cones, they actually have to be hit pretty directly with a photon of light in order to pass the signal forward to or anteriorly to those ganglion cells. So the photons of light have to come all the way back to the back of the eye, to the retina, and not only just to the retina, but all the way to the back to the most posterior structures of the retina, which are the photoreceptors, and then the information is sent forward through a series of different physiological effects. Now, I have to always choose with a truncated lecture like this, I always have to choose which pieces of information I'm actually going to be discussing. So we're not going to be discussing phosphodiesterase and the changing of the pigments and so on, and how some things are inhibited and some things are excited and so on. For now, just in a really general sense, we'll say that the photoreceptors receive a signal, they're passed forward to the bipolar cells, and then those are passed forward to the ganglion cells. Lastly, I wanted to come back to this larger picture of the entire eyeball and talk about emmetropia, myopia, and hyperopia. The emmetropic eye is an eye which is shaped just correctly enough that light hits exactly where it's supposed to hit, all the way back to the back of the eye to where the photoreceptors are waiting for those photons of light. However, there are relatively common conditions out there that are worth discussing quickly. A myopic eye or a myopic eye is an eyeball that is too long, and therefore the light tends to hit a little bit in front of, or sometimes quite a long distance, depending on how long the eyeball is, tends to hit in front of the focal point that is meant to be hit at the back of the eye. Now, the myopic eyes are going to be corrected with some kind of a concave lens, a lens that dips in the center. As opposed to hyperopia, and hyperopia is the opposite problem. You have an eyeball. The main cause of hyperopia is an eyeball that is actually too short, and therefore the light is going to be focusing behind, if you will, behind where the retina exists, and this would have to be corrected with a convex lens. It's also worth noting one other general feature of the eyeball is the way that it can change when objects are going to be closer up. And so we're going to be starting to discussing the concept of closer up. And it turns out that 20 feet is what we call the far point of vision, where anything closer than 20 feet, you need adjustments. For example, you need to be able to accommodate your lens. Accommodation of the lens is where we take those suspensory ligaments in the ciliary body and we actually call it a bulging of the lens so that we can better refract the light. Next, we have pupillary constriction, where we can actually shut off some of that scattered light. When we constrict our pupils, what we're doing is we're taking the pupil and making it into a smaller hole. Constriction of the pupil makes a small hole in the middle of the iris, and therefore less light is being scattered through, and it allows for more clarity in that way. Lastly, if something is quite close to the face, oftentimes we have to converge our eyeballs, which means that we're actually bringing them closer toward the center and kind of crossing them toward our nose in the direction of the nose in order to be able to see something that is quite close to our face. So accommodation, constriction, and convergence are all aspects of when we're trying to see something closer than 20 feet. It's actually harder on our eyes, believe it or not, to see something that is closer up than it is to see something that is farther away, because our eyes do have to make these kind of adjustments when something is closer up.